thanks for the invite, and thanks for the all the support here with the materials that we've developed at Rutgers. Hopefully that'll offer some greater promise for you all in the future with regard to your cranberry operations. What I'd like to do today is maybe give you a little bit of a history of where we came from and perhaps where hopefully we're going in a positive way, that is. The cranberry is quite unique, but most of you know it's one of the few fruit crops that's native to North America, blueberries being another fruit crop, muscadine grape being the third. A lot of people in the U.S. say American is apple pie, but does anyone know where apples were domesticated? Huh? Close, Tajikistan. So they're an import, but cranberries is truly a North American crop. It's native to southeast Canada, northeast U.S., and the industry basically started back in the mid-1800s with varieties that were taken out of the wild. Early Black, Howes, McFarland. I believe Early Black was selected from a bog in Massachusetts in 1843, and it's still grown today in Massachusetts and somewhat in New Jersey. But I think you have to look at it from the perspective those varieties that came from the wild, and that also includes Ben Lear, Lemunion. I don't know if there's any Lemunion being grown here. The plant essentially was there, not for us, but basically it was surviving, and it was a population, the cranberry population that was adapted to the acid soils, and had an option either to produce fruit or produce vines. As opposed to apples, you plant an apple orchard, you come back 50 years later, the tree is either big or perhaps died and is no longer there. Cranberries have this alternative method of propagating. It's called asexual propagation, but and that's what we refer to as the runners. And so there's this constant, I think, battle or decision making by the plant whether to produce seeds through the fruit for really the survival of the species in the future. Because what the seeds do is they generate, just like most biological populations, including animals and us, is that there's many new gene sets that are put out there, and whatever's as you heard, the survival of the fittest for that particular climate, for that particular environment, survives. And if that variation wasn't there, probably most species would die out. So cranberry, before we came along and started to grow it, had its own needs as far as survival. And runnering is one of them, and to some extent we're dealing with that today. Probably many of you know that a lot of the varieties that are grown today are actually mixtures of perhaps a variety that wants to produce more fruit, as opposed to those like Barrenberry in Wisconsin and Searles beds that doesn't even produce a flower. And so over time, the variety Barrenberry will muscle out typically the variety Searles, and these beds start losing productivity over time. When I first started at Rutgers, I had this notion that nothing changed. I went out to McFarland beds out in Washington, in Wisconsin, and then you start hearing terms like, well, this is a true McFarland. This is a McFarland that doesn't produce. So and then also with Stevens as well, and we're still dealing with that today, because the cranberry can produce runners, but we also exploit that for propagation. So, getting back to breeding and genetics, the first essentially genetic improvement that occurred in cranberry occurred in New Jersey, and the reason why it was all predicated on the fact that we had a disease called false blossom. Thank God you don't have it here. That as the name implies, it's a phytoplasma. It's a bacteria-like organism that doesn't have a cell wall, and it devastated the New Jersey. Actually, New Jersey, I believe in 1880, had close to 12,000 acres of cranberry. We're down to about 3,500 today, but in the early 1900s, it went down to 2,500 acres, and the disease essentially took a lot of cranberries out of production that never came back. And the first breeding program was government sponsored by the USDA, and you all are benefiting from it today because out of that program in the 1930s, they made about 40 crosses, produced about 13,000 seedlings that were planted out at White's Bog in New Jersey, and the varieties Stevens, Pilgrim, Wilcox, Franklin, and Bergman, which many of you have here, and Beckwith came out of that program. So that was essentially the first breeding and selection cycle for genetic types that would be more suitable for cultivation to grow the fruit. You have to look at the fruit from the standpoint of the plant. It's actually a stress on the plant. It does nothing for the plant. It just takes nutrients away from it, growing, runners and vegetatively, and being vigorous and competing with the weeds. So what you're obviously we're not interested in eating a cranberry salad, but the fruit is a load on that plant. And so that was the first breeding cycle where they put out about 13,000 different genetic types, and out of that came out Stevens, and Pilgrim, which are still very popular today. So that was the first breeding and selection cycle. And back then, what were they selecting for? Color, yield. The other interesting thing, I think, is back then in New Jersey, most of the soils were the cranberries were in muck, which is highly degraded peat. And in New Jersey, I don't know if you've heard this year, but we're into our sixth heat wave with temperatures over 30 C or over 95 degrees. And what happens is that muck releases nitrogen, and varieties like that were selected out of the wild, like early black and Howes, start to runner and produce almost no flowers and produce only runners. So in effect, what they were doing is they were selecting for varieties that would use that nitrogen a little bit more for fruit production. And so that was the first, I would say, genetic improvement for varieties that will produce more fruit with higher nitrogen environments. And I sort of use that in my breeding program, and it's actually not, it's not any new news, because for example, corn, which has had a lot more genetic improvement on it through the years. The other thing with corn is that it was domesticated 10,000 years ago. It's an annual, so the corn has gone through about 10,000 generations of genetic improvement, whereas cranberry in the 1930s was the first cycle of breeding and selection for agriculture. And out of that came more productive varieties and I forgot to mention that the whole purpose of that program was to develop a false blossom resistant cranberry. And the other element or component to false blossom is that it is transmitted by a bug or an insect called the blunt-nosed leafhopper. And what they were actually doing is they were doing cafeteria tests where they would take 5 or 6 varieties like McFarland, Early Black, Howes, Searles, and put them in potted plants, put them in a cage, and put blunt-nosed leafhoppers in there. And they would, and they found out that the blunt-nosed leafhopper did not like Early Black, I believe, as much as the other cultivars. So they felt that if they bred a variety that the leafhopper did not like, then the spread of false blossom would not be as fast. But thank God pyrethroids came along, and essentially the control of false blossom was done by controlling the leafhopper with the pyrethroid insecticides. But out of that program, I would say nothing came out that was false blossom resistant or leafhopper that repulsed leafhoppers. But out of that program came the varieties Stevens and Pilgrim, which are still grown today. Now, back then, it was largely, there was no juice, there was no SDCs. It was all for jam or sauce. And so they would do pectin analysis. They would take these lots of fruit, and they would determine how well they gelled. And so that was the criteria that was used back in the 1930s and 40s. Then came along juice with Ocean Spray, and so a whole new market developed, and so the criteria for what was good for that changed. I really never assessed our varieties for gelling quality because the principal products today are SDCs and juice. So from the standpoint of breeding and genetic improvement for the 21st century, our criteria of what we need for the current for the current needs of the processing industry have changed. SDCs, as I understand it, are the principal or major outlet for your fruit. And as probably most of you know, the criteria for what they want from the standpoint of the quality of the fruit that they're getting has changed. So fruit firmness right now is on the list. That probably not. That criterion really is basically only surfaced in the last 2 years. The size of the fruit for the SDCs, certain sizes are required. Early black and Howes or small fruited back in Massachusetts, early black only gets to about 1 gram. In New Jersey, it's about 10% bigger, about 1.1 grams. But as I understand it, the size of that small size is not suitable for the current process for SDCs. So, and back in the 1930s with Stevens and Pilgrim, fruit size wasn't really a criterion, but I think it contributed to the increase in yield. You could have the same fruit set, but if the berry size is 2 grams like we have in New Jersey for Stevens, you essentially doubled the crop with the same set. So these are, the point I'm like to make is that things are changing and the criteria are changing. In New Jersey, we've had the most warm, I would say hottest, you could say the warmest years in the last 2 decades. And Stevens was selected in New Jersey in the 1930s when things were cooler. And it turns out that D. Moranville and Crimson and Mullica, which were selected in the 90s and in the early 2000 years, we found out have a little bit higher heat stress tolerance. I'm not talking about the fruit, I'm talking about photosynthesis, the plant photosynthesizing. So we generated this through crosses and hybridization, populations of plants that had different genetic backgrounds. And we were selecting in an environment when things were hotter, and then, so by just naturally, we were selecting for a new combination of traits that basically were adapted to our current climate, our current soils, our current management. And essentially that's what breeding is. And so we are in our infancy as far as genetic improvement. Stevens was the first breeding and selection cycle. D. Moranville, Mullica Queen, Crimson Queen, Scarlet Knight, and then of course the Wisconsin varieties essentially represent our second breeding and selection cycle. And those were selected largely in the 90s when juice was still the criterion. So you all were getting a higher premium for the higher the TACI, the higher your returns. Now that's changed. Now we're looking for the SDCs a window between 25, I believe, and 40 TACI. And the other thing is that with the juice, you can take a berry that's 100 TC and one with 0 TC, and you basically got 50 TC. Well, that's no longer acceptable because you're not making a juice, you're making SDCs, and so the criteria now has changed that every fruit has to be 50 TC. You can't have any variation. Well, that would be the ultimate criterion that we're breeding for. So certain varieties, like I believe Haynes, has a much more uniform coloring pattern, whereas with some like Stevens, you'll get white fruit and you'll get dark fruit. So the varieties of the future are essentially going to continually, I think, have to be selected for that are appropriate for the current climate from the farmer from that standpoint. The one problem right now with the SDCs is the fruit firmness. Although I think environment, which is your harvesting methods, how much water you use, how long the berries sit in the water, probably have a huge impact on the ultimate quality of the product for SDCs. We can start selecting for varieties that maybe tolerate sitting in water longer than the ones we currently have. We're into our third breeding cycle. There's crosses out here that we planted in May this year that essentially are largely Mullica Queen derived. So, for example, Stevens has the what we think are the best genes from Potter, which came from Wisconsin, and from McFarland, which is a native variety from Massachusetts. The Franklin variety was a cross between Early Black and Howes. And so De Moranville, for example, here, which was a cross I made in 1988, is a cross between Franklin and Ben Lear. So that one has a lineage from Early Black from Massachusetts, from Howes from Massachusetts, and Ben Lear from Wisconsin. Mullica Queen is quite unique because it's a cross between Lemunyon, which is a native from New Jersey, and also from a cross with number 35. The number 35, as getting back to our first breeding program, the USDA breeding program in the 1930s, they put out 13,000 progeny from about 40-some crosses. Out of that came Stevens and Pilgrim, but there are also other selections. They came up with 40 selections. And apparently this was number 35 selection, and it's a cross between Howes and Searles. Well, why wasn't it named? It was probably not named because it's very late and has very poor color. And the poor color, basically, I think, is what is why they never named that one as a variety. However, it was kept in the New Jersey experiment station, and we had a trial in what we called Bog 17 with Stevens, Crowley, Pilgrim. There's 8 other varieties, Early Black Howes, and we measured the yields in that bed for over 10 years. Number 35s out-yielded Pilgrims and Stevens over this 10 years, but the fruit was smaller. So the program that I utilized, and Haynes and Welker and Mullica Queen all have number 35s as a parent, and that's a cross between Howes and Searles, between Howes from Massachusetts and Searles from Wisconsin. The number 35s out yielded everything there, and with a smaller fruit size. So it was producing more fruit and more yield, but it was doing it differently than Stevens and Pilgrim. So I started utilizing that in the breeding program as a parent, but it had lousy color. And I was fortunate when I crossed it with Lemunyon, which is a native from New Jersey. We grew out about 150 progeny from that cross, and it turned out that one of the most productive seedlings from that cross had the second best color. So Mullica is not as early or as high colored as Crimson or De Moranville, but it's actually a little bit ahead of Stevens. And so here we combine the genes from New Jersey, Lemunion, with Howes from Massachusetts and Searles from Wisconsin. So it's a quite unique variety in that has a bloodline from 3 different growing areas. So as we look into the future, we with these trials that we have out here, these aren't seedling trials, but we're trying to put out different genetic backgrounds here to test under your environment, your conditions, and your management techniques to see what's best suited for you all here. What was kind of interesting is that one of the siblings of Haynes, which was a cross between Crimson Queen by number 35. Now, Crimson Queen is a cross between Stevens and Ben Lear. So Crimson has Potters, McFarlane, Ben Lear, and crossed with number 35s, it has Howes and Searles in the background. So it has 5 of the original wild selections from the wild lineage in that plant, and it's done extremely well in the trials in Wisconsin and also in Oregon. Although Kim seems to think that it's declining, the problem is that when you're, as I mentioned earlier, when the plant starts producing 300 or 400 or 500 grams per square foot, the whole nutrition program has to be changed to accommodate that the amount of resources that are taken up by the fruit. So as I mentioned earlier, we have a learning curve with these new varieties. All the work that's been done with nutrition has been done with Stevens and Early Black and Howes. Well, I think that whole mindset has to be, you have to open up your minds as far as to see how these are going to handle it, particularly when the crop is, when the variety is putting so much work into the, so much of the resources back into the fruit and not into the plant. So we've changed the whole dynamic of what the plant wanted to do to grow vegetatively to what we want is to produce more fruit. So I think I'm going to leave it at that. I'll be happy to answer questions if there's any out there.